Today we will be having a challenging task of playing with clocks of times, and we need technologies for this. So the first one will let us see all these 133 slides that I have, and because I will have to rush through it, so it will be a little bit superficial and not as technical as some of you want to, but I will give you some time afterwards. Let's get started. A few words. You can keep it open. Let people see that. No, make it open. Yeah, we are having open source approach, and actually one of the things that I'm doing, I'm active open source developer. I'm also doing a lot of other stuff like gene therapies, like bioinformatics, genomics, transcriptomics, and of course aging research and some other scary for some people words. But today we will focus on the major task of turning back the clocks of time. And to focus on this task, we first have to look into our present moment. So here we are, actually having fun here at Øredev. It's a small subpopulation, but I believe that it's representative. You also guys have fun. And let's imagine that what will happen in 30 years. I believe it's a great conference, so it will still exist in 30 years. And what will happen if no progress will be in biology field? So probably we will look like this. And we will not look as fancy and nice as we are looking now. And honestly saying, probably we will actually be pretending to be as happy as we are happy now. Because in reality, like the honest state of us is that we will have some pain. Our brains will not be as bright as they are now. We will have some disabilities, so we will not be able to process information as good as we do now. And the reality is that for thousands of years, we couldn't do anything about aging, and we all lived in this sense of internal terror. Something bad will happen. We cannot do anything about it, so maybe let's just smile and pretend that it doesn't exist. Or maybe not to feel bad all the time, maybe we should invent some excuses why aging is natural, everything that is natural is good, so maybe it's okay that we are feeling bad and dying. Also, COVID is also natural, and for some reason we are not happy with COVID and flu. Or maybe we can come up with excuses that we don't want to overpopulate the planet, so we have to die and feel bad. Or maybe some excuses that we don't want to get bored, and maybe if we will have some sclerosis and dementia every day, we'll feel like something new happening in our life. So even though it's ridiculously sounding excuses, actually it's what happens so often that there is a whole terror management series that explains why so many people rationalize that something that makes them feel bad actually is good. And the thing is, I believe that if we really want to be as happy in 30 years as we are now, maybe it's nice to be in a good health, so to have some biological progress to at least be like this we are now. Or maybe some of us would want to have some additional health like we had in 20s, to have some time for extracurricular activities. And the thing is, so we need some technologies to deal with the damage that accumulate over the years to repair it. We also need to understand, because it's not like a magic, tomorrow you are young and you were old, that we have to somehow measure if we are doing well or not. So here is a lifespan curve, also known as Kaplan-Meier's one. It's actually the most robust but the slowest way to measure. So here you can see that on the top, if we have a population like that of the same age, like 100% is alive in the beginning, and with passing of time, part of us start dying until we get like maximum species lifespan and everybody dies. And what we really want, we can actually play and extend our lifespan. What has been happening in the last century, mostly we were just curing diseases, so we had the shape effect that we could just survive for more, although it didn't improve our overall health a lot. And in the end, we just got so burned out that we are not dying from one disease, but we are dying from another. Because if we cure diseases separately, aging and deterioration still exists. And what we really want is to actually have the scale effect to move the curve to the right. So our maximum lifespan will also increase, and our health will also increase this way. The problem is, and actually why we need more sophisticated methods, is that if we will apply this approach to be happy in 30 years, probably we would have to have like experimental and control human groups that will age for 130 and more years, and probably it's not realistic to wait 120 years to actually know if the treatment is working. And for this reason, we actually need faster methods to project. If I am doing tomorrow a little bit better or a little bit worse than I am doing today. And also, we have to know how actually we can measure not some specific disease, but aging overall. So here is a short context of my presentation. Going from aging itself, looking into the biomarkers, looking into the machine learning models that actually can make these predictions based on short time changes if our treatments are going better, then also understanding the complexity of aging and finding ways to do cool stuff like turning humans into whales to live way longer. Whales can live more than 200 years, just for those of you who don't know. And also going from explainable AI to the mighty AIs that can explain stuff to us, and having like this longevity genie helping us with our research. And if we start from aging itself, so aging is a deterioration and decrease in viability with time. Here you see diseases, how they exponentially increase with aging. And here you see accumulation, all types of damages like DNA damage, like extracellular matrix damage, mitochondrial damage with age that we have to fix somehow. And when we have this trajectory that over the life we get to our prime in 20s or 30s, and then we have pretty bad scenario. And of course, we can do some things like better lifestyle or some simple treatments to make it slower. But ideally, we actually want some radical technologies to fix aging faster than we age, to reach so-called longevity escape velocity, like rocket escaping from Earth's gravity. And first, we have to actually look what has been achieved. So first, we, of course, experiment with animals. So for C. elegans, it was possible to extend lifespan 10 times. For mouse, because it's more complex animal, overall 100% extension was possible. So maybe we can do it something for humans as well. And but first, we actually, to go from mouse to humans, we have to have an ability. For mouse, we can like just wait until they all die, but for humans, we need those biomarkers. And biomarkers, they are divided into molecular biomarkers, all like omics data, genomics, transcriptomics, biomarkers of organs and tissues, how your liver doing, how your lungs doing, and functional biomarkers, if I am doing well, if I am feeling well, if I can run well, and if I can function well. And also different biomarkers, it's harder or easier to explain their values to make changes depending on type. So actually it's so I had a paper on it, so with the review you can check it out. And going like quickly through it, so for functional, here is for example the sports records, how they change with age, so you see clear deterioration. Here, for example, what is close to me is marathon running, so you can see that it gets very slow around 80, and I recently did my Hello World Marathon, so I feel a little bit bad, so you see like red, I am pretty bad in this, but at least I am better than 80 years old people and their world records, even though they had time to train. And here is what happens with our intelligence, long-term memory, working memory, pretty much everything, with exception of ability of doing blah, 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 like politicians do. So if you are a politician, you will get better with time, despite aging. And for those biomarkers, so here I talked about functional, and if we talk about physiological, usually it's harder to interpret, but usually what you do, you train machine learning models that try to predict your age, and in case if, for example, they say you're younger than you are, there can be two cases. The model makes a mistake, or you actually really biologically younger. And it's the main challenge in aging, to train those aging clocks, those machine learning models, that they should show you're younger or older when they talk about biological age, and to differentiate it from cases when they make a mistake. There are different techniques to do it. You can analyze historical data with disease incidence, with people dying, and the model should predict, for example, premature aging people, like people with progeria, that they are older than their chronological age, and vice versa for people that live longer, but it's still not that easy, and you need a lot of data for this. And there are also different modalities, so this is tables from my paper when I reviewed different types of biomarkers. So this is the easiest that you can measure even now, and actually can do it for free. It's your blood biomarkers, and you probably do regular blood tests. They are not the most accurate, but still, like R² can be 77% in some cases. Also, something you can do very quickly is with the photo, facial clocks, even though it more tells about your skin rather than other systems of organisms. Then there are also other things like 3D aging clocks, like fMRI clocks, and different clocks focus on different issues. Like for brain aging, probably MRI head clock is the best. And also, if we talk about what we know about genomics, transcriptomics, proteomics, we can also have clocks with this. Most of you know about genomes, about DNA, so this kind of strange, but in reality, apart from DNA or genomic sequencing, we also have a lot of other molecules like RNA sequencing, transcriptomics, we have chip sequencing, we have methylation sequencing, a lot of it. And here are, I'll just go quickly through it. So for transcriptomic logs, here are examples. The most accurate one, some of you know, is methylation clock. But unfortunately, it's very hard to explain it. So you know that you are, for example, I am 4 years younger with methylation clock, but I don't know why. And I don't know what to change to improve it, because it's very hard to explain, actually. And the different clocks, they show different aspects of our health. That is a challenge. And there are also some other types with immune, microbiome, and so on. And this is what we know about the clocks. And as I showed you here, that they explain different aspects. And what you want to change your lifestyle. So you need some explainability. What should I change to get better? And for this, we actually use explainable machine learning. And what the problem is that there are two types of models. White box models like linear regression or, for example, decision tree. We can more or less have idea what is happening inside, but those models are not the most accurate. The most accurate are often something complicated things like neural networks, that it's totally black box. You don't know why it makes this decision. And to cope with this problem, DARPA suggested this idea of explainable AI when you have a special type of model. So you have your own model that makes prediction, and you have a special model that uses the first model to explain how it makes prediction on what influences it. And the problem here is that many like neural network models, they have different reasoning than we humans. For example, here is a famous one pixel attack that you change the one pixel and you can turn a sheep into car, deer into airplane, because some visual models, they make decisions very differently from humans. Same with the noise attack. Two same pandas for human, and then panda and gibbon for the model. And to cope with it, we have our Black-Scholes very precise model, and we apply this model as a tool. We permutate the input and use explainable model to actually explain how different factors influence it. So there is like many different ways and technologies to do it. Like here is the whole stack that you can check. For images, explainable models, for example, they can highlight that, okay, we predict that there is a problem, here is the region, so you can look closer what to change and where things are located. Here are some popular technologies available, here is that become really popular recently. And the ones that I used a lot was shapely additive explanations. And in explanations, there are actually two levels. The first one, what is important for us here is like, it's not mine. Actually, on a blood aging clock, I think it's accurate for me for chronological aging. But if we look into this, what you can say is that you have a prediction, and here on the X, you see different parameters of your blood formula, and you see like your chronological age, and each of these parameters, it moves prediction to higher or lower levels. So here, for example, you can see that BAN or total cholesterol, they actually contribute to higher biological age prediction, so probably you have to work with them. Yes, that's how you can reason about your own predictions. But if you want to look into how the whole model is doing, you need the predictions for the whole population. So here is SHAP values plot. And here what you can see is that the idea is that you have the base, so like 0 is an average for the whole data set, average prediction, different factors, different features, they move the prediction to like higher or lower biological age in our case. They kind of shift it, and the colors are like red is high value of a feature, blue is low value. In some cases, some features like you have high cholesterol, probably it's bad, so it's correlate with higher biological age prediction, but some parameters can actually be vice versa. And we used actually this technique in some of our papers. So here is a little bit about whales that can live more than 200 years. And if you port some of DNA repair genes from whale cells to human cells, they become 50% more radiation resistant. So there are some potential ways what we can learn. So we did some work comparing different species. And the idea is that long-living species, they may be unrelated, but because they optimize for longevity, they find similar ideas, like how to improve your DNA repair, for example. And here is an example of things, how it happens in nature, like shark, dolphin, and the ichthyosaurus. They have very similar shapes because they have to adapt to water, but it's like totally different species, really unrelated. So we had a pipeline that we use multiple models and a bunch of samples. I will go quickly through it. So usually we talk about big data when we talk about bioinformatics, but actually it's kind of lie, because even though like a raw genome can, for example, be 400 gigabytes, in reality you have very small number of samples, but the dimensionality, like for example, you have 40,000 genes. And in the study, because we looked into orthologous genes, we had, I think, around 18,000. So you have several hundreds or thousands samples, and you have tens of thousands features. So very wide data, but very small. And for this reason, we use a bunch of methods. And actually, neural networks didn't perform well. They just need more data for high feature samples. And we looked, we had simple linear models. We had Shep value with light GBM tree-based models. Here, I will not get into it, but you can see some ideas how typical explainability charts look like. And here is a decision plot, like you have, it will converge to one point, like an average model prediction, and each, on top you can see like what is the lifespan, like for human maximum lifespan, like 123, I think, years, and you can see trajectory for that for each sample. Each gene, it shifted the prediction towards higher or lower. So here we had not age, but maximum lifespan. The better, the larger, the better. And you can see that for some species, some genes, like for long-living, DRK gene had the best contribution. Yeah, so you can maybe read on your own on more details how it works. Here are some other plots, and here are some integration of data, and we got some genes that ideally we haven't tested in the lab. We didn't have time and funding, but maybe if we will genetically edit humans with those genes, maybe we will, or maybe we should start with mouse, we can extend lifespan. And also there are problems with causality and other questions. And another thing that I want to cover is actually what we can do with more static information, like our genomes, that say if we will live, if we have chances to live longer. And also it's a kind of t-shirts problem. So some of you ask what all those genomic things that I have on my personal report on my t-shirt. So here we also have some explainability features. There overall, in my case, I started the project because I didn't want to be a shoemaker without shoes, like bioinformatician that cannot analyze himself. So it's an open source project, and let's say, and everybody can do it because sequencing is really cheap. You can sequence yourself, but there are no really good solutions about longevity, and we developed an open source library, and we have the largest in the world collection of genetic variants connected with longevity, actually. So you get a tube, you put your saliva, you send it to any sequencing provider like Nebula or Dante you want. Then you can get their results, but you can also run our open source library on your laptop and get some reports. The problem here is that there are like 600 million known genetic variants. And here are, for example, some of the traits that show heritability of different things, including IQ. And here it's very hard to interpret because it just increases or decreases your chances to have some phenotype. It's not always straightforward. And one of the techniques that are used for explainability is connecting information, like we have a neural network and we connect information with known biological pathways and we change the topology of neural network to connect it with biology. It is kind of biased approach, but at least we can say which biological processes are connected with some gene variant that shows that has contributions to a better phenotype, like longer living or some less risks of a disease. And yeah, so it's some data that we had. Unfortunately, heritability is only like 15 to 20% for aging. So most of your chances to live long is your lifestyle and medicine, with exception of extreme longevity, where heritability is higher. So more than 100 years, probably heritability plays a role. And some intermediate conclusion from explainability is that if we understand what is happening, it gives us the power how to change our treatments or lifestyle to improve the parameters. It also allows us, like with examples of some genetic targets, have some targets that we can address because the model reveals that this gene is important for longevity of a gray whale, for example. And we can build new therapies. And the other aspect that I want to cover is that actually we have explainability AI techniques, but it would also be nice to have almighty AI to explain stuff to us. And with all those large language models, we would like just to ask AI, what should I do, how to tackle this problem, to like help us. And this is one of the projects that we are developing called Longevity Genie, is a set of open source libraries for better usage of large language models with biological data. And we were, of course, inspired by this huge growth of large language models. Like you see that ChatGPT got 100 million users just within several days. And it's a bit outdated data on sizes. And one of the things that we wanted to actually start prototyping our solutions is just understanding of those convoluted genetic reports. Some of them I have on my t-shirt. And you can see that gene variants, green is good, red is not good. And it's pretty hard to even like for trained people, because there are so many genes, to actually make sense of it. And for these reasons, we wanted the large language model to actually answer what this gene variant is about. So we had a baseline, we asked ChatGPT, and we saw that for popular genes like ApoE, it somehow manages to make some answers, but for the less popular the gene is, the less chance it had it in training data, and the higher chance that it will hallucinate and tell us wrong stuff or just general stuff. Here is an example of true and fake answers. And there are some problems with logic and reasoning with LLMs, even though AI, people think they should be logical, but it's not the case. And we tackled it, so there was this event, Zuzal, like 2 months event, combining living together, working remotely, AI, longevity, crypto, network states, synthetic biology, and there was a hackathon. And at the hackathon, we actually addressed these problems, how LLMs can make better answer, how they can actually reference what they know. Because usually ChatGPT doesn't say how it knows, just says, I know that this gene is this, but where did you take it? And here is just a slide how typically you plan the development. And by that time, we didn't have opportunity to fine-tune the model, so we used techniques that we orchestrate. So we have this large language models, and we use just ChatGPT API, and we just submit it with additional data from databases that we obviously have and from scientific literature. And here I selected, I have some nice FOXO polymorphisms, and I wanted to know what they mean. So like native ChatGPT, it gave mostly useless information. It's like a bad student. I will tell whatever I know about the gene, but I don't know anything how it influences longevity, but at least I will tell something. And also by that time, we also had Longevity GPT as a member of our team, Nikhil Yadala, so it can ask literature, and it also uses ChatGPT API, and we got a bit better results. And then, so there are some limitations of it because it was mostly literature-based and API-based. And we wanted to have some Swiss knives that can not only have like literature asking like papers, but also can deal with databases, run some agents to answer the questions, and can also deal like with your personal data. So that's how the project was born. And we had a chatbot at the hackathon, and we added some databases, and you see we had some better results, like something green, more meaningful. And the way how it works, so we used, it's called like complicated way, retrieval augmented generation, but in essence, it's very simple. You have like a query of a user, and you don't send it as is. You first make a search in all sources what you know about the topic, and you send both query and additional information to ChatGPT or Llama or whatever you're using. And as it has both additional information and your query, it can do better. And the way how it's done, so it's done with in-context learning. So all those LLMs, you can see that they are just decoders that you have some initial text like user query, and they just generate the next token, next word, each of the step. And if you give more text with some additional data, so they can take it into account into generation and get smarter answers. And also in their representations, for us it's words, but for them, they have like vectors, so they vectorize all the words. And because they are trained, similar words like apples and bananas are closer. And actually, this is heavily utilized with so-called semantic search, when you can just use this knowledge and it will find similar things like similar genes that are connected with your gene, or similar, I don't know, fruits if you're asking about fruits, by just using this semantic search similarity. For this, people use vector databases. We also use them. And yeah, so we use different embeddings. Surprisingly, the ChatGPT one was not the best one. There are open source that are better specifically for semantic search, and you can find it on Hugging Face. And yeah, so here are like some of our methods like papers and other stuff. And here are examples that we are getting better results. And the ChatGPT doesn't know. In our case, we have some specific answer. Also similar example. And also we started using not only RAG, but we started also using agents. So what is agent? Agent actually is a way that you can use large language models to be an agent that interacts with different tools and data. Now, ChatGPT also uses agents to browse the web, for example. The idea is that you call LLM, here is the question, tell me what is required to be done, and it tells what to be done. Then you can parse the output, and you pre-instructed which tools it can be used. So you can just find it in an answer and just run the code and return the observations of those tools. So it's called React framework, not the JavaScript one, but the idea that first it has a thought how to solve it, and it has all the menu it can do. Then it generates JSON that we can call functions to give additional data as observation to the large language model, and then we give this extra data, and we ask LLM to have a thought, are you confident you know the answer now, or we have to do more actions? And how it is was, so here is a log. You can see that it asks what is longevity effect of GG variant, and in the log you see that it's entering agent chain, so it has a thought that to answer this question you need the RSID information tool. Then it generates JSON, and my app, it intersects this JSON, and technically this JSON is parameters of a function. As an observation, it gives results, and then knowing the results, LLM says, oh, you know, the weights are pretty high, like 0.38, so probably it has moderate longevity effect. And there are also some tools to have UI way of dealing with those agents and LLM calls. And as a developers, actually, it's for you, it's super easy to implement it. So you use frameworks like LangChain, like a Python framework. And here you see a function, a tool function. And here's the use tool decorator that parses your comments. And those comments, you actually see that you have more comments than the actual code. So the comments is an instruction to ChatGPT or Llama or any other model you are using, how to use this function and in which cases. And the function itself, it can be pretty simple. And you just write a lot of these functions. That's how you help it with agents. And yeah, more complex agents where you ask about clinical trial results. There, actually, ChatGPT generates SQL code to query the databases to get what you want. There, it's a bit more complex. You have to explain it, the structure of database and how to deal with it, but it's more flexible. And yeah, so here are other examples. And another use case is that in some cases, instead of just querying databases, you can ask it just to generate code to answer. And most of mathematical answers, like in ChatGPT, actually, large language models, they are bad in math, but they can generate Python code and Python is, if you use numeric libraries, it's good in math. So that's how it's usually done, code-based agents. And of course, we have to talk about safety. Now we have this nice prompt injections to generate some crazy code. And what I found fascinating is that in all those science fiction, they say that to protect you from evil AI, it shouldn't be able to modify itself, it shouldn't be able to run code, it shouldn't be able to get extra information, it shouldn't be able to trick humans. And here, what reality we have now, what companies try to enable AI doing. So it's really interesting. And another thing that we didn't do, but we are starting to do now, is fine-tuning, technically additional training for large language models with additional data sets. And now it's possible to make it efficient. You don't need huge hardware to do it, because there are techniques of parameter efficient fine-tuning when you actually use quantized models. You can look into Hugging Face sets like with rounded numbers. And also you can use techniques that you train adapters, additional matrices that you apply on top of a model. And you don't have to train like all say 90 billion parameters model. You just train small matrices that you add on top of model, and they can be also interchangeable and tunable. Yeah, so here is the code that we have. We are actually adding even more stuff there. And so far, it may sound a bit complicated, but actually, most of the stuff that we do, it requires just general software development expertise. You don't need to, you learn like several concepts, and then you are ready to go to improve stuff. You don't really need deep biology understanding to contribute. And what we are focused on, so we had some data what users want, so they want to know more about recent studies, they want to ask more about aging, about drugs that they can take, about personal health strategies. And in case of scientists, probably the most people who would benefit the most, they actually need some way like workflows when LLMs will, for example, open some papers, do reading for them, suggest some drugs from databases. So it's more complex use cases, but it can save a lot of time and move the science forward. And we are actually, it's our major priority because we want to speed up the progress in biology overall. And yeah, so here are some things that we plan to improve. And yeah, some hybrid search, some agents, some fine-tuning. So here's some preliminary results, what we got better. Like clinical trials, drug-gene interactions, longevity drugs. Well, we started doing fine-tuning, and actually we're now in a stage that everybody can contribute. And we also believe that if we make it open, that community can step up and make it even faster. And that we can also fine-tune to make it better even than ChatGPT for our use case. And we can also use open source LLMs for many cases. If they are fine-tuned, they actually can perform better or be cheaper and more flexible. And here was the team, and here are some QR codes. And I believe that we have this goal of being happy in Øredev 2053. And we have some tools that can help scientists to speed up the progress. And well, everything is open source, so let's make it happen together. And thank you for your questions.